Hello, everyone, and welcome to this episode of Gen Live entitled How Lab Automation is Advancing Biotech. I'm Juliana Lemire, Deputy Editor-in-Chief at Gen, and I'll be your host today. So lab workflows are constantly evolving to handle higher throughput demands, increased efficiency, and more complex tasks. For some, that means having automation play a bigger role in the lab. Whether talking about one liquid handler or a modular system of multiple instruments, the role of automation in streamlining workflows is increasing. Many agree that automation affords scalability and offers more accuracy in lab work. However, there are also challenges in the automation field as well. On this episode of Gen Live, we will discuss the role of automation now, the ways that it is affecting things like efficiency, cost, reliability, and also we'll talk about what the future looks like for this field. To do that, I'm joined today by two amazing experts in this area. First, we welcome Rachel Cohen, the Director of Plate-Based Analytical Assays and Head of Automation at Prime Medicine. We're thrilled to have you here today, Rachel. Thanks for joining.
Thanks.
And also joining us is Emma Norris. She's a startup strategy consultant and former strategic partnerships lead at Automata. Thank you so much for joining us today, Emma.
Thanks, Juliana.
And before we jump in, I want to remind our audience that we would like to answer your questions too. So please put any questions that you have for Rachel or Emma into the Q&A section of the platform, and we'll get to them as soon as we can. So let's dive right in. Awesome. So first, I want to take a moment to learn more about you, Emma and Rachel. I hate reading bios, and I just think that you can do a better job telling us kind of about yourselves, and also really curious to learn how you got into automation. So Rachel, let's start with you.
Yeah, so I've been at Prime now for a handful of years, 3, and started my career off at Merck. I think a lot of people in automation kind of come across it the same way I did, which is I stumbled upon it. I got hired as a contractor originally doing assays, and turns out the lab that I got hired into happened to be a high throughput analytics laboratory. From there, I loved the robots and I loved tinkering with them and kind of just progressed through there to now head up the team at Prime.
Okay, very cool. Emma, how about yourself?
Amazing. Great to hear more about your background, Rachel. I also started as a scientist in the lab and realized pretty quickly that I didn't want to pipette all day long and I wanted to spend more time doing higher value scientific analysis. So I started off my career in AI for drug discovery at Lab Genius, which was working at the time in its infancy in drug discovery. And we had this age-old problem that data in equaled data out. And I really wanted to try and find a way to solve this quality problem and create clean data for AI for the future. And that's my journey into lab automation.
Okay, awesome. So many different experiences here too. Rachel, I want to get back to also being at Merck versus, I think we should talk about automation in different areas, whether it's kind of a bigger pharma drug development or Prime Medicine, which is a smaller genome editing company. So, but anyway, I'm getting ahead of myself first. And I should say, people who watch Gen Live know that, I have a PhD in microbiology. So I was a lab scientist, so really far away from automation. We didn't use any automation, but I was really I went to SLAS, the automation meeting, for the first time this January. And I was so enthusiastic. I walked out of there just feeling like, oh my gosh, this was so exciting. I really didn't know entirely what to expect. The energy, the enthusiasm for automation, it was just really, which is actually what prompted me to want to do this particular episode. So I think so I just want to say to the audience too, I'm really just learning about automation myself. So what I want to start by doing is defining automation, because I think it's maybe harder to define than I might think. So I guess I'll throw it out to either of you. Can you how do you define automation?
So I think I would my stab at it would be that it's anything where we're moving a process from totally manual kind of to something that's automatic, which I realize is using the definition as part, or like the word as part of the definition. But if we think about manual processes, especially in the lab, you get plate washers. So for those in the audience who have had to run an ELISA manually by hand, instead of adding liquid to wells, you get a robot to do it. Instead of moving a plate around, you have a robot do it. So it's automating those like minor process tasks that other people would generally do manually.
Yes, it's a really interesting question, Julia. And I think that the way I've often thought about it, particularly in my previous roles in sort of partnerships and developing relationships with labs, both in pharma but also in industry and smaller biotechs, I've thought about it more as using solutions that leverage tech to solve pain points in the lab. So even more broader, how do we just free up scientists' time so that they can focus on the high value problems? And what that does is it often helps us think about those core pain points as opposed to the things that we think that we want to solve. Like we think we just want to, reduce the amount of time that it takes to write up our experiment in an electronic lab notebook. But actually, maybe there's something further to the core of what we're doing in our processes that we want to try and solve. So that's often a way that I like discussing it with customers and clients in the past.
I like Emma's definition much better than mine.
I wouldn't go quite that far, actually.
They're synergistic. Very good. And so you've both been in this field for a while. And like I said, I mean, my introduction is a lot more recent. So having watched this field evolve over the last, say, 5 or 10 years, where would you say the field is today? And I'll say I have one rule on the show. You cannot use inflection point. Because that's definitely what people use. And I feel like fields cannot always be at inflection points, right? But anyway, that's if you want to use it, it's okay. But would you say it's like still in its infancy? Is it maturing? Where are we at?
Rachel, do you want to kick us off on this one?
Yeah, sure. I think if I compare the lab very specifically lab automation spaces to other places where people have seen automation, like manufacturing facilities, people always show videos of cars being made in automated manufacturing. If you look at it in that scope, we're pretty early on. We're very far removed for how those industries are kind of where they are at currently. And we came into it later. I think people had bigger problems to solve before they wanted to move a plate or wash a plate. So we're there, but it's also growing pretty rapidly. When I came into the area, there were only a handful of vendors. None of them wanted to work with each other. So you were kind of shoehorned into their solution, and maybe they were really good at one thing, but not really good at others, but they made products across the board. And we now have kind of a community growing where there's interaction and there's free knowledge sharing. So we're getting closer to the other end.
Yeah, I couldn't agree more. I think, again, thinking about the definition of automation, you have all the way on one side from a sort of standalone individual process, a qPCR machine or a washer, to end-to-end, effectively like production lines, like you would have in a car manufacturing facility. So very little human interaction involved in a process. It may have uncapping, it may have washing, it may have liquid handling steps. And then, we now see Gemini robotics in the last 7 days coming out. And I look at them, and I think, goodness me, there's a world where a humanoid coming into a lab is possible. Mm-hmm. And I don't think many of us would have seen that over the last couple of years. So, where are we at? I think we're really early days, but there's so much going on in the space. And I think we can leapfrog. I don't think we necessarily have to go stepwise in the way that we automate labs. We can actually, like the mobile phone did in some countries in Africa, we can do the same with lab automation. I'm really excited to start seeing that happening.
Yeah, I mean, walking around the SLAS floor, you would think there's so many different robots and vendors and companies and it seems like definitely the technology is growing so rapidly. I guess one thing to talk about is adoption. Oh, but before we do that, actually, I wanted to say, Rachel, I really like your point about like the automotive industry, because that was exactly the point. I did a fireside chat at SynBioBeta with John Brennan Vidal, the CEO of Opentrons, and he was making that exact same point, which is like compared to other industries, we're really far behind. It's just that it needs to kind of take off in the biotech and the life science industry, I guess. Which I thought was an interesting way to think about it. Well, one question before we get to adoption is why do you think life sciences is far behind? I know, Rachel, you said people, and this is true, are worried about other things, but like, I also feel like maybe people just love to old-fashioned pipette. I mean, is that part of it?
They do. Oh, they do. I think people are a little afraid because you take the control away from people when you put it on a robot. And oftentimes we see, like, if an experiment goes wrong, people are very quick to then be like, oh, it's on the robot. That's why the robot screwed up. And we can pretty confidently say the robot usually doesn't screw up, almost never. They program to do the one thing over and over and over again. That's their job. But people don't like not knowing and not seeing everything happen. And we have that. I think there's also, because of how far away we are from the automotive industry, there's a skill set that is required that isn't, there's no school currently. There's no degree sets. Most people don't see this stuff until they get into an industry, into a lab that happens to have it. And what often happens, and I'm sure Emma has seen this as well, is you walk into a lab and there is a robot. There's a liquid handler sitting in the corner collecting dust because it's never run, or the person who ran it left and didn't train anybody else. And so there's a knowledge gap there that we're working on building out. And that's definitely a problem with the adoption.
Yeah. Maybe just a couple of ads. One of the things we often have seen in the past is people think that robots are going to be magic, right? And they're going to solve every single problem under the sun. That's obviously not the case. And I think the paradigm that robotics and automation is going to enable and help humans to better do their work versus completely replace is one that's starting to come into the lab arena. But we did often, we started off with this concept of usually an arm in the center with machinery and devices around the arm, which wasn't flexible. You, as a scientist, you don't recognize it as a normal way of working in the lab. And we're starting to shift towards an environment that looks like the normal lab setting, perhaps even with robots at the back of a system, so you don't necessarily see them, and you as a scientist can walk up to your devices in the same way that you'd normally do. You can do your analysis or your low throughput assays yourself manually, and then you get your robot to do the high throughput assays. So there's, I think, a real paradigm shift in the way that we think about automation as augmentation as opposed to replacement. And that for me is going to completely change the adoption curve over the next couple of years.
I think just to kind of piggyback on that again, the industry is also seeing a shift in that, when I first came into the industry, it was build the biggest system you possibly can. It can be as high throughput as possible. So you end up with these monstrosities of equipment that take up, tens of square footage of space footprint wise across giant labs. So they need tons of space, and you have to interact with all these different types of pieces of equipment to actually use it. And there's a barrier to that. And I think we're pendulum swinging back in the opposite direction, where we're smaller systems now. They're doing a thing. And if it's an autonomous robot or a human being moving things between systems, that's more common and more asked for than the giant, this can do everything all at once, but if it breaks, 17 people have to fix it.
Yeah, that makes a lot of sense. I mean, you have to work with the scientists, right? Not just say this is what you're going to do now. This is this huge tectonic shift in your work.
Yeah.
I mean, adoption, people get used to what they've done for many, many years, right?
Yeah.
Okay, I want to touch on other challenges in a little bit, like I know cost and things like that, like all these barriers to entry. But first, I want to talk about going back to what we were going to touch on, adoption. Where do you see adoption? I mean, would you say pharma, biotech, even academic core labs or academic labs? I mean, where would you say automation is really taking off at this moment?
I've seen it across all of them. And it totally varies depending on the backgrounds of the people. Often the key is like, is there an automation scientist currently in the team? If yes, like they're thinking 5 steps ahead even than sometimes the vendors themselves in terms of what they're looking for. And that isn't necessarily correlated with the startup community versus pharma. I've had startups who I'm working with who don't even know what a washer looks like, right? Like they're just totally used to being in the lab from academia, and using manual. So the profile of the people that one's working with, at least from a vendor perspective, completely changes the way in which these projects tend to run. And a lot of often what we have to support and do is education, to try and help the scientists understand like what actually is this going to do, how does it work from an engineering perspective, so that they can get the trust, as Rachel was mentioning, to be able to walk away and say like, yeah, I'm happy that this is kind of a black box, but I'm willing to know what the science is doing inside it. So yeah, I'm not sure that was a very good way to answer your question, but have seen it across the board, and it hasn't necessarily been correlated with people doing AI versus older school labs.
Yeah. And I would echo that. I didn't spend a lot of time in an academic lab, but we know that there are academic labs out there that have automation. Clearly, a startup, especially in the Boston biotech area, there's a large community of Boston lab automators, for those who are not aware, and that most of us are at startups. So there's a large startup presence for lab automation. Big pharma also is very much into it, to varying degrees that we've seen.
Yeah. The one other thing I just mentioned briefly, I'm based in London in the UK. We've seen people like the NHS starting to get really interested in automation. There's a huge problem with cost, and the expectation on turnaround times, particularly on things around genomics and diagnostics, is going through the roof in terms of expectations. And the only way to solve that problem is automation. But that has proven harder to actually, really get into the NHS, particularly now with NHS England no longer existing, which was, for those in the US, a national body basically who assesses tech and innovation. And NHS England no longer being around makes it a lot harder for the innovators in the UK to try and bring great tech into the NHS. So a real shame for the next sort of 5 to 10 years.
Okay, got it. Yeah, I would imagine some of these barriers are harder to get over for certain. I don't know why I think academics would be a little bit harder than like a company. I don't know.
I mean, the sheer cost of automating things it's beyond kind of a lot of other things. You can get something that's not automated, like an incubator, for
right
tens of thousands of dollars. The automated incubators are hundreds of thousands of dollars.
Right.
So there's a scale there, and in an academic lab, that's not really a thing that they're going to want to spend money on. Be paying people or doing whatever.
Yeah, interesting point. Also, Emma, what you were saying about the genomics, because it's like, as I cover omics a lot in my writing here at Gen, and like as the throughput in just even DNA sequencing gets higher and higher and higher as it is, and the sequencing cost comes down and down and lower, like those are hungry machines to feed, right? And like you're saying, probably the only answer to that is automation, really. I mean, I know that automation and genomics have gone together for a couple of years now, pretty tightly.
Yeah, and I mean, what's really interesting specifically in the sort of pre and post PCR space is you can have lots of different types of automation. So you have, on deck, you can use a TCAN or a Beckman to do like a large range of processes, or you can off deck everything. And so there's a huge amount of thought that can go into like, what are you trying to optimize in terms of your metrics? And that differs, like depending if you're a cancer hospital and doing diagnostics versus you're an institute who's effectively a service organization for the rest of the institute. You're really going to be thinking about different things. And usually that's flexibility. It's the classic flexibility throughput way up. And, I think that's the real challenge, at least for me in my work, is making sure that we fully understand what the customer's looking for from those metrics. Often they think that they want something that they don't. And that's the major challenge, because once you set up automation, often you're limited by your hardware to move things around later down the line.
So it sounds like, really thinking through what you need before you start is pretty important, right?
Yes.
So, Rachel, so when you joined at Prime, how much automation was there, and like has that been built up a lot under your tenure there, or like how? I don't know if you're comfortable speaking about that, like, so personally, but like if you could walk us through that, I think that'd be super interesting.
Yeah. So when I started, Prime knew that they needed automation early on. We're a gene editing company, so therefore we do a lot of sequencing. We do a lot of genomics.
Right.
So before I got here, I think Prime was a company for like 18 months prior to me arriving. They had a Beckman that was doing library prep PCR2 for sequencing, and they had purchased another system, a large integrated system, to try to help meet the need. There was not any other piece of automation, plate washers, readers, what have you. When I showed up, we now have, we're up to 17, huzzah, integrated automation builds that kind of vary between size, use case, purpose, teams, and location across Prime since I've been about the 3 years that I've been here.
Wow. Would you say that that's like a typical, like, that seems like a huge expanse very quickly? Or do you think that's what's happening?
So it is a huge expanse. I think there was at Prime, there was a need, and there was support from our leadership team. I came in on the technical development and operations side. So our CTO, the first person she hired was the VP of quality. The second person she hired was me. So she knew the importance of automation and how critical it was, not just from a throughput standpoint, but also to control variability, repeatability, like precision and accuracy for assays, which was how I started here originally. And it just kind of expanded from there. When you have robots and they're kind of producing and showing good data for a variety of things, people inevitably want in to some extent. And that's kind of how it grew across the board. When you have your leadership team who supports it, it makes it easier.
Interesting. I've also read that automation is kind of becoming popular in more clinical applications. I think largely because the notion is that automation would increase reproducibility and accuracy and things like that. Do you agree with that?
Yes.
And I mean, does it not make mistakes though, ever?
So I think at least from an equipment side of the world, they do make mistakes, but the mistakes are crashes, right? They're like, generally speaking, hardware, software errors. You don't have, they have tracking, and the clinical ones have a lot more tracking than necessarily we do. That will track, I scan this barcode, I put it in this well. That's a programmatic thing. Those things don't, generally screw up, which is better traditionally than a scientist who has eyeballs and brains and thoughts kind of outside of the lab at any given point in time, who isn't sure what well they put stuff in. But if it's loaded incorrectly, if it picks up something weird, you start to get errors where that can propagate and cause bigger problems. But they're definitely like different baskets of issues that happen.
I totally agree, Rachel. I think that the one thing that is interesting here is there isn't that much data that looks at processes that have been done manually versus in an automated fashion. And that's a real shame, because I think there's tons of opportunity to look at how much benefit there could be from automation. We did something with a customer around QC metrics, and it did show significant benefits in a PCR process. But it's, that's a small amount of data that I'm going off there. But generally, we also saw, you mentioned ELISAs. There was one customer who would have to repeat around 50% of their ELISA processes, sometimes even up to more like 80. And with an automated process, that wasn't needed. So the huge knock-on benefits of that, not just from quality of data, but also sustainability, lab consumables, time spent in the lab, ability to turn around test results faster is huge. So I think with quality also comes some of those other benefits that happy to speak sort of more to as well.
And we've, just to kind of go off of Emma's point, when we start to automate a process, we tend to use at least whatever data that process has as like the benchmark for manual to automated, and we will run the comparisons. We generally see that automated brings down your CVs. It's, your spike recoveries get better, your pipetting is more accurate. It's kind of all of those things. And then we can track it and get people doing more scientific work with their brains than manual repetitive processes.
Got it. Yeah, that makes a lot of sense. Okay, I'm going to ask our first audience question, because they're coming in fast and furious. So this is from John in the audience, and he asks, what is the split between processes that can be automated versus those that cannot because they require more variables to change, the throughput is small, or some other reason? Okay, so maybe and then there's a follow-up question, but would one of you like to tackle that question, or both of you?
Yeah, sure, happy to. Thanks, John, for the question. Yeah, it's a great question. So throughput being small is often a really big problem around cost. So that is the main barrier usually that at least I've seen in that space. I'm trying to think of processes where throughput is small, but there's really high value to results. I think there's some opportunity there. The other thing to think about in this space is creating automation hubs. So this is something that we're seeing in a lot of pharma, where they're creating massive labs, where it effectively is an automation service, where any scientist from across the site can come down and build their own workflow using all the machinery available. And that, with great software, hopefully leads to a world where you can be flexible and you can start building these workflows, at the drop of a hat. In the past 2-3 years, workflow writing has taken anything from a couple of days to a couple of months at times for really complex workflows. Mm-hmm. But we're moving towards a world where that may not be the case. So no code solutions, drag and drop solutions. There's even some really exciting AI, so free text to SOP and then to automated SOP. So there's real progress in that space to try and speed up the workflow creation step, which at that point I think will give a huge amount of flexibility for people to use automation for slightly lower throughput or lower value sorts of tasks. I don't know, Rachel, does that correlate with your experiences today?
Yeah, I will say the only thing that I maybe disagree with is that there are some low throughput applications that are, I guess, high value that we do automate, and specifically because of the world I live in, it's GMP analytics. Mm-hmm. So GMP analytics to do like product release generally have a sample or two. So that's a plate in my world largely. But automating those processes, even if it's only for an hour, is better than somebody else because of the control that we get around it. I think oftentimes we end up with things, and Emma had touched on this early, that, the data, the way the experiments are and the way the assays are set up, or the processes, if it's not a good process, the robot doesn't make it magically better. If the assay is not good, or variable, super, I don't know, challenging, difficult to do, it doesn't make the robot, they don't do it any better than humans at that point. So if we often get requests like, this assay is, it's not performing well, the robot will make it better is not the answer to that. And that's usually where we kind of stop. The other thing is when steps of the process are just not automation friendly or automatable. My shining example of that is we got asked to automate an assay where they were mixing tubes, but the mixing of the tube was dragging a tube down the side of a tube rack. And this motion is not really a really fun automation problem to solve when we have other automation problems to solve. So things like that, when people don't think about how those processes actually translate, it becomes that's where our line tends to be crossed.
Yeah, totally. And maybe just picking up on that, I think that is one of the main reasons we've seen much more automation in biology than in chemistry.
Yep.
Is for exactly that reason, in terms of sort of complex processes. We need to swirl this in a certain way at a certain frequency. Like that's actually quite difficult to do with robotics, and probably over the next couple of years. I mentioned Gemini earlier, but with improvements with robotics, we'll be able to have more flexibility in terms of what we do. But at the moment, most of the robotics or the arms are just moving SPS plates. So unless things are able to be in the SPS format, it makes it largely quite difficult to automate.
That actually came up, Rachel. So I should say that the way that I was introduced to you and your work is because at SLAS you gave a talk. And in that session, I don't know if you remember, probably maybe not, because you were the talk right after him. So maybe you were.
Oh, James.
Yeah. And so he was talking at Novo Nordisk about using some AI to measure the liquid levels.
Yep.
Some of and so that seems, correct me if I'm wrong, it falls into this category of a place where automation was kind of really hard to develop and design to work in their workflow, right?
Yeah, and there's a really cool paper. It's less about he was using the AI vision to figure out levels to determine how much volume to pour out of a bottle. In a similar vein, there's a paper that was published, I think, a year or two ago, with someone using AI to learn the laminar flow of fluid out of a bottle to be able to pour your standard Gibco bottles of media into a flask, but with a robotic hand instead of a person. So there's a spot where these things are starting to happen, but there's some that we're going to go with easier than others. Racking of a tube, I will live in the force feedback on an arm does not really interference.
Yeah. But it's good to appreciate, right? That these things are actually super hard to do.
Yeah.
I mean, I think that's
what
I got
about it
from his talk as well. This took a lot of effort to be able to do this. So, okay, terrific. One other question actually also from John. So thanks, John. Because we were just talking about academia, maybe having a tougher time and it's so expensive, which I want to talk about in a minute. But the question is, will automation exacerbate inequalities in biotech and academia? The rich are going to get richer. So what do you think about that?
I think in some cases, I'm going to say with the caveat of with a good automation team and good people running the instrumentation and vendor support, you can do faster with automation than you would do generally manually. Mostly because robots don't care what time of the day it is. They don't need lunch breaks. They don't need coffee breaks. They can just kind of go. But it requires a team around them to support that. I don't know if it makes the rich get richer, and I saw, but it does help with that process.
Yeah, it's a really great question. I'd never thought about it in that way. So thank you, John, for bringing that up. I think perhaps not a today thing, but in the next couple of years, I expect to see more decentralized labs that have robotics. I mean, we've seen cloud labs already, but more of the CROs and those types of organizations are definitely going to automate themselves. And in that world, hopefully the cost of lots of these processes comes down, and academia can leverage the robotics elsewhere. But yeah, I do think that there'll be a world where the biotechs and the pharma have access to great robots and can do fantastic throughputs, and your PhD won't be able to in a normal university. And how we solve that problem, would be really interested if anyone has any suggestions as to how to solve that moving forwards.
And what about Emma? I think that you win the prize because, well, we all do, because I think we went actually 30 minutes without saying AI, which I actually think is totally just
machine learning
needs to be called out. We didn't say AI, but we have to. So
we're going to say AI.
So, I'm just going to ask you, what is the role of AI currently? And how do you see AI going forward in automation? It seems like these two are inextricably linked.
Yeah, so there are lots of ways to answer this question, and maybe forgive me if I answer it in a couple of ways. So AI in automation for me is really around drug discovery.
Yeah.
And it is using automation to enable the world of AI. And we see literally, startups coming out of SF every week who are doing drug discovery, and they are all automating to facilitate that. So that's sort of slightly separate from the automation. Actually, how is AI helping automation? We see it across. I'm thinking of examples like OpenTron's new AI assistant, to Briefly Bio. What are they doing? So OpenTron's is an interesting one. They're using AI to allow you to effectively use free text to say, oh, can you actually, change this particular part of the process? Or actually, I want to prepare this amount. And that's kind of really interesting that they're using it to do those, what I would call quite low impact processes. But I think it's an entryway to start using AI into these processes. And it's a really great introduction. Again, Briefly Bio is doing something similar. So that they're taking SOPs, creating it into an automation language and using AI to effectively help you mirror your or connect your SOP to the automation system that you have set up in the lab. And the AI creates lots of suggestions. Every time that you make a bit of an error, it will suggest and fill in the gaps for you. So AI at the moment, at least from where I'm standing, is doing a lot of those like low value add, but kind of helpful to help the traditional scientist who might not be as experienced in lab automation to feel more comfortable in the space. And so that's really key. The thing that I'm excited about for AI in the future is how AI can influence our schedulers. And so when we're thinking about full end-to-end production line automation using loads of different devices, one of the key issues is how do you schedule each step of the process to ensure that you are not breaking time constraints or effectively breaking the science that you're wanting to perform on each particular plate that you're going through, that's going through a system. And AI, I think, will be able to solve those scheduling problems a lot better than we can. But that may require AGI. That may be more of a sort of, depending on if you agree with Dari or you agree with Demis, a 2 to 5 year timeline for AGI, and then plus another 5 years after. But I think that's where I really think that interface between automation and AI will play.
Okay, cool.
I think on our end, we have from like an automation engineer standpoint, so not the low hanging fruit, but the maybe somewhat higher level hanging fruit is that we can do things now with AI like give it SDKs or software development kits and APIs for our software, assuming the integration companies and the instrumentation give us those, and have AI like help or assist in writing drivers to connect instruments together, which lifts a burden off of engineers, especially when we're doing things in a DIY fashion. So we have kind of that going for us. The other thing, and it's far more kind of not super globally helpful, I'll admit, but we gather metrics on all of our systems now, all of our pieces of equipment, to get kind of like how far they've traveled, what's gone wrong. And we can start to do things with AI machine learning to like predict when things are going to fail from a service standpoint, to try to get there before the arm falls off the robot, which I have seen on more than one occasion while it was running, to try to beat us to that point so that catastrophic failure doesn't have to happen and we can be proactive in our services. That's where we're kind of seeing it from an automation engineer standpoint.
Okay.
I'd love to chat with you about that afterwards, Rachel. Would love to hear more.
It's my shining thing.
Yeah, I have a question actually about like integrating different automation systems together, because I mean, I guess the question is, how hard is that? Like if you have a system that has, you know, everything from a spatial platform to an NGS to a centrifuge, I mean, is it, how hard is that to do that? Is it?
It depends.
Okay.
I think that the scheduler is the main piece of the pie at that point.
Okay.
So there's a handful of scheduling companies kind of on the market that all have kind of pluses and minuses to them. But what we see often is, especially in like a startup biotech space, is money constraints of asking for new equipment to be added on can become kind of cost ineffective. Timelines that are gifted to us from those scheduling companies, if we want to integrate devices, can also be cost ineffective. So we've taken on, and I know a lot of other people in the field as well have taken on the integration ourselves in a DIY fashion, which is difficult, but the pieces are there of the puzzle to kind of get it in, and you can get very, very good at it if you have to. But we don't have a standard kind of language that all of these pieces of equipment communicate with. They've tried with SELA as like a communication standard, but that didn't go, I think, as far as everyone wanted it to. So everyone's unique and different because we're all special. And so getting special drivers for each and every piece of device that communicates on its own unique wavelength is definitely like a challenge. We're better now than we used to be. The field did not used to have anything. So we're better off now, but we're still getting there.
Perhaps slightly jumping the gun to what excites me for the future. Maybe am I allowed to do that, Juliana? It's
Sure. Yeah.
You know, we've been in a world where the biggest variability that you see is based on who's done the experiment. Like, did Rachel or did myself do it? And you know, the data looks totally different. We're now getting to the point where, depending on which automation system you're using, and the devices on that automation system, your data looks different. What I'm excited about is the world where actually you can send the same SOP to loads of different labs across the world, and they all have different devices, all probably have different robots and different schedulers, but you can actually get out results and data that you can compare. And that world is a long way away, but it will make a huge difference to the way that we do biology.
Yeah.
Is another challenge, I mean, going back to the omics world that we were talking about, and the, you know, higher throughput is so exciting and for so many reasons, but also creates its own challenge now of data analysis and, you know, kicking off just like terabytes of data at the same time. Is that something that is a problem, or a challenge rather, in automation? Just, you know, higher throughput in general, and how do you deal with that, Rachel?
We automate the data analysis process too.
Okay.
You kind of can't, in a high throughput world, you cannot get one without the other, because you've just moved the bottleneck. And so we have automated genomics processes. We have automated like potency analysis platforms to do assays, ELISAs. So with a click of a handful of buttons, you know, you get data results out that's QC'd and, you know, everything else. But at least in my head, there's not one without the other. You need both.
Okay, so
can't second that with much more. Totally agree.
So something for people to think about, if there's people in the audience that are thinking about automation, I guess that's kind of a good point to be also considering, is the data analysis side of it. Back to our audience questions. So Wouter has not a question, but a remark. This is, I think, going back to, I think he put this in earlier in the program, but it says automation needs mind switch, and you need to foster that carefully in your lab with your personnel. I think that was like when we were talking about the challenge of kind of training people and needing to, have that kind of adoption. Okay, he has another question. How do you process your data? Barcoding, scanning, data architecture, an interface to analyze that data?
Yeah, so we live in a world where everything's barcoded all the time, always. We will even contract with companies to get barcoded labware so that we don't have to barcode it ourselves, so they don't come pre-barcoded. We have barcode scanners and everything to track everything that's going. They export really fun CSV files at the end that are swept up and processed via some form of code and pushed back into Benchling, which is our ELN or ELN right now.
Okay.
And the one thing I'd say there is, we've seen a lot of automation that doesn't need complex barcoding, doesn't necessarily need complex data pipelining. Like, you can do this all in a vacuum effectively, and just know exactly where you've put the individual plates. So, some of these qPCR processes might have 2 plates for some of the customers that are looking for automation, and that works really well. It's just when you start getting to high throughput that having the full end-to-end process to help you with quality and to help you with just making sure that you know exactly what data is coming from what plates. Because trust me, when there are robots moving things around, it's easy to not know which plate is coming out.
And then to scale that, if there's 384 well plates, 1536 well plates, now you have thousands of data points that can come off a plate. So knowing exactly what's in any given well at any given point in time is kind of critical when you start to get to the high throughput scaled up versions of this.
Definitely. Okay, cool. Another question from the audience, from Thomas. Do you see a future where automation allows for experiments to be moved from the minimum of triplicate repeats to duplicates? This would save so much time, money, and precious samples.
Yeah, I think a lot of industry, I don't generally run my things in triplicate, I'm going to be honest. We do dilution series for our assays, but we are very much in duplicate. We have good control of the equipment. So yes, I do think that that's a place where you're headed, if not already there.
Okay, cool. Okay, yeah. So actually, Meg has an interesting question. I don't know if you know of any companies where equipment for automation is provided as an in-kind contribution for like a large research program. Meg, I'm not sure how to answer that. But I guess.
I think
go for it. The one thing I've seen, Meg, is with a couple of institutes, they've done projects alongside suppliers to prove out some new process, or using it on an automation platform where that particular device hasn't yet been integrated. And in those cases, we've often seen sort of strategic partnerships working really well. Whether or not that's something that you're able to do with the devices you're speaking about, I'm not sure, but definitely worth kind of having that discussion, because people tend to be really interested in having their names on a new white paper around automation or results if they're new in the space to it.
Yeah, I would agree with that. We've definitely been a part of, or I've been a part of different collaborations or things where you automate something with someone and they, you get a discount, you get whatever is like payment for it.
Okay, that's a good place to start. Thanks. Emma, I think I'm going to throw this question to you. What has been the most interesting out of the box use case that you've encountered?
Oh, goodness me. Well, definitely unbottling and taking the tops off bottles to have beer. We've seen people do that. Had there was one particular robot in a lab where they'd got an updated robot, so they had this robot sitting on the side, and it was people's like evening activity just to play around and see what they could get the robot to do. I say that all in jest, but application-wise, it's largely around having a full end-to-end process that's AI lab in the loop. So where you have your AI system both designing your experiments in the first place, you then run it on automation, and then that same model effectively suggests changes or suggests iterations to the way that you run those assays, and you iterate it's like within this box, and those have been the really exciting projects to work on. As Rachel's been talking about, it includes everything, including data pipelining, data analysis, and those have been the ones that have excited me the most.
Rachel, how about yourself?
Oh man, I feel like I don't get to see it's mostly like my own crazy brain. So I don't think we've ever been complimented on our out of the box thinking. We do a lot of I think we get excited about just building out new instrumentation at this point, which is and getting new arms in, because we've started kind of collectively moving forward as a lab to not the standard robotic arm that you probably saw literally everywhere at SLAS. So
yeah,
this arm. It's everywhere, white labeled for any company that wants it. We're moving away from that to more industrial 6-axis arms to kind of try to push the boundary on the manufacturing, kind of going to automotive manufacturing. And we've gotten feedback about that people are excited that people are adopting like industrial arms as an option now.
Are there there's a question in the questions in the Q&A. I mean, one thing I think that I've learned a lot in this hour, but one thing I don't think I really appreciated is how, and this is actually how we kind of started out, how like people kind of fall into automation. Like there's not a training program, as you said. Are there communities in areas where like automation people can kind of get together and compare notes?
Yeah, so I'll hit on that at least a little bit. So the SLAS is a very large, the Society of Laboratory Automation and Screening is a very large community. They mostly do yearly events. They do have like other smaller events, but like the big international conference is the one that we've been talking about. There is another, they're an interest group, the Laboratory Robotics Interest Group, which is more localized. They have a New England chapter, they have a Philly chapter, kind of Reno, San Diego. So those are smaller communities that meet on a somewhat regular-ish cadence, where people kind of exchange ideas. And then newer to the thing, and if Sam might be here, hi Sam, if you are. Sam Muller started the Boston Lab Automators, which is kind of, it's local to Boston, but it took off in a couple other places. So I think they have a San Diego Lab Automators, there's a Philly Lab Automators. There's actually a Munich Lab Automators, which are more casual events. Vendors come and sponsor it. We get to see some robotics kind of up close and personal, and it's just a good time with people to kind of discuss what's going on or what you're doing, and share ideas. So those are kind of the three big ones, at least right now, that I'm aware of in the area.
It's awesome to hear about what's going on in the US. It's very clear
Come hang out with us sometime.
How much further ahead the US is on this. In Europe, it's still like relatively early in terms of bringing groups together. Obviously, we have European SLAS, as well as then going over to Boston, San Diego. And there are also groups like, if you type in things like London Automators, if you're based in London, I think there are a couple of others in the rest of Europe as well. You'll often find groups, largely people who are on the ground kind of sharing experiences and thoughts and code. And those tend to be quite useful.
Okay, awesome. I want to get 2 more questions in, so they might be like a little bit of a speed round. But this is a great question, actually, kind of based on what we were just talking about from Nathaniel. How would you suggest academia better prepare students for this increased automation in the workforce? And how would you suggest someone new in the field get better acquainted with automation to make them more desirable as a candidate for hire? I mean, students, it's going to be so big when they are coming out in, looking for jobs.
So we at Prime, we have automation co-ops. So these are positions that are 6-month like internships that are open to students at varieties of universities in the area to kind of learn with the automation team here, which is a really good way for students to get in without having to make the academic purchases to actually build up the infrastructure to have that as a learning opportunity. Programming is clearly like already a thing that's happening in universities, so that's helpful to them. But I think that type of experience is probably where it's better suited. And I'll speak for Prime, but we had this experience at other places and across the community as well, where we often hire people who are fresh out of college because there's not a lot of experience. And we enjoy teaching people.
Right. Yeah, so maybe it's just really familiarizing yourself if you're, a younger student with the industry and what can be done. Yeah. Okay, that's terrific. So now it's time, Emma, for the future, future predictions. So, I mean, what do you see? And I guess, what do you see over the next 5 years as being, what do you think we'll be seeing in 5 years? Where do you think we'll be? I guess that's an open-ended question.
Where I think we'll be maybe isn't where I hope we'll be. Maybe I'll start with where I'll hope we'll be. Very much a world where lab to lab, you've got connection within one organization. So this is already in lots of the Basel labs. You have tracks connecting all of your different laboratories. So humans don't even need to walk from one laboratory to the next. All barcoded, as Rachel's been talking about. So end-to-end systems that really take a sample right from the start of a process right to the end. And that might include 5, 10, 15 assays in the process. That's a world I see for big pharma. The world that I see for startups is using much more flexible automation and robotics. And often that's because with startups pivoting, they have to have this ability to not put too much capital up front on a very specific system, but using more of the opentrons like robotics and automation to support a whole range of different processes. What excites me, I mentioned it briefly, but I think maybe relevant bringing it back is, the real challenge that CROs have today and lots of labs is that they can't compare data that's done across various different laboratories. And that prevents CROs from dealing with peaks and troughs in their workloads. If they validate a process on one system in one lab, they can't spread it elsewhere. And I think the more that we automate, and therefore the more that we understand operationally what is happening to a sample, like what is the actual process that this device goes through, operationally, we'll be able to compare and contrast that data from multiple different labs. Very much the dream. I don't think we'll get there in 5 years' time, but I think we can be on that journey to start to get there.
All right. Well, I'll have you guys come back in 5 years and we'll see if your predictions are right. Rachel, just in like 30 seconds, sorry, or a minute, any future predictions?
I think I'm excited to see the cost go down, not gonna lie.
Yeah.
We need that in the industry more than I think a lot of other things. Things that we saw at SLAS that I think we're going to just continue to take off are things like vertical integration, which we had not seen previously because space is a limiting factor, and modularity. So the ability for scientists on their own to flip and flop equipment is going to be probably more local impact, but huge for the industry as it currently stands.
Okay, awesome. What a very exciting space to watch. Well, that brings us to the end of our session of Gen Live. This was a lot of fun, and also I learned a ton. So thank you both, Emma and Rachel, for for educating us. And please, to our audience, join me next month on Gen Live, where we'll be talking about gene delivery and gene therapy. And now I'm going to ask them how they're using automation. So I'm going to bring it forward. With that, I'm Juliana Lemire. Stay safe. I'll see you next month on Gen Live.